we do have a next session coming up, which is scientific editors. It's kind of unusual. I'm not really familiar with cases where you could get a panel of scientific editors together. So let's find out what they have to say.
Well, thank you very much. Pleasure to be here, and we're thrilled to have editors from Nature, Cell Metabolism, and Nature Metabolism. George Caputa, the senior editor of Nature, Rosalind Mott, scientific editor of Cell Metabolism, and Christoph Schmitt, chief editor of Nature Metabolism, all highly relevant, of course, to our area. So I'd like to kick it off by asking you to comment on what kinds of steps, especially as scientific editors, can be taken to help move the field from a therapeutic focus to a health focus, to a preventive focus. And I'll follow up after you comment, and I'll throw it open to all three of you.
Sure, I can start because I'm unmuted. First of all, thank you for the opportunity. This is a really unique meeting. I love how there's multiple stakeholders. It makes it a very thoughtful discussion. I've enjoyed it a lot. But I guess sort of broad terms of moving things in a different direction, because, you know, as editors, we see this sort of comfort zone of C57 black 6 mice and sort of a formula for doing metabolic studies and, you know, knocking out this gene and then doing established metabolic disorder and then targeting it with a pharmaceutical. That's sort of the formula that we see.
Of course.
But I think that, you know, this meeting and conversations like this are really important to sort of to change the culture around that, right? I think a big thing really is just changing our relationship with time, right? So humans evolved to sort of think about things acutely. That's been important in our survival. But it's really about shifting our mindset, and this has to be proactive. It's hard. Our mindset for chronic thinking, long-term thinking. So the big example I can give that we're really struggling with is climate change, right? It's something that slowly evolves over time, and aging is the same thing. So motivating people to work on that problem that doesn't have this acute sudden effect on us is just it's a mindset change.
Excellent, thank you. Before throwing it open to both Christoph and George, I'd like to ask you a follow-on question, Rosalind. You mentioned the sort of standard approach, C57 black 6. What, if any, approaches do you think we take towards translational misfires with so much reliance on the black 6 mouse?
So we're seeing more of the genetically diverse mouse panels being important in work, but also, you know, doing your mouse work, but then relating that back to human studies. You may have discovered a target in mice, but if you look at human genomics, does that target make sense for the condition or disease that you're studying?
Mm-hmm. Thank you.
Yeah, so I wanted to follow up on your second, on what Rosalind had to say. Yeah, C57 black 6 mice are really, you know, kind of, as Rosalind said, like where people go first when they're doing metabolic studies. But the honest truth is they're mice, you know, and there's millions of years of evolution between humans and mice. And I think more and more studies come out every day showing, in terms of metabolism, how dramatically different mice and humans really are. And I think that might be contributing to a lot of the failure of maybe basic academic scientific discoveries to really translate into relevance to human disease. Maybe not human biology, but human disease, I would say, in human health. And so I think people being more cognizant of that, obviously a lot of these organoid models, Rosalind said, like the diverse genetic mouse panels are incredible and excellent for kind of, instead of just looking at one strain of mice, we look at multiple, but also moving to more human tissues and more humanized kind of animal models and tissues are all ways that in terms of doing and performing science, we can make things more relative and maybe try to get a better idea in the preclinical space if what we are asking and the system we're working in could have relevance to human disease and human health. Going back to kind of the first question, though, I would say that, at journals, we see kind of a diversity of different types of studies. We see basic science, we see translational science, so like the mouse models, and then we see preclinical data, so things that are really prepping for these clinical trials in terms of maybe primates. And then, more and more we're seeing actual clinical data published in our pages. So nature metabolism, and cell metabolism. And so, all of the pieces of that pipeline are really important to get to a discovery that is going to impact human health and human disease. So, we can't cut out the basic science for the translational science, because a lot of things that are found in basic science translate into clinical discoveries. But I will say the one thing that I have noticed that has really helped in terms of kind of refocusing researchers into thinking about problems differently is kind of this ability, at least I notice in the United States, and it's not so much in Europe, unfortunately, of people jumping around and kind of taking different roles. So PIs working at a pharma company, or people like people doing a PhD in molecular cell biology, but then maybe working in epigenetics or working in an NGO. And it really brings diversity of perspective, and you really start to learn about the kind of hurdles that there are in all of the different fields, like was discussed in the previous talk. All of the different hurdles that, maybe a researcher personally, when they're throwing a compound on a dish of cells, probably aren't really thinking about. I think we get this a lot at Nature, where people propose new treatments for all different types of diseases, but the real question is like, does it work as well or better than what we have already? Is it cost effective? Is it going to be accessible? Is it cheaper? Is it easily mass produced? Is it easily administered? Those aren't things that researchers really are thinking about, and maybe it isn't something necessarily that they should. I guess that's a question for everyone to think about. Like, when should these questions about practicality really come into play? At journals, we're really just here to kind of lift up and show everyone what's going on in the field, and hopefully inspire people to kind of move in a direction. So.
Thank you. I'd like to invite Christoph to come into the conversation.
Sure, thanks. Yeah, there was a lot to unpack here, I guess, already. So I maybe just going back to the original question as to what we can do in terms of, focusing more on prevention rather than treating disease. Yes. And that's it. As journal editors, there's not an awful lot we can do, I would say, other than being open-minded and actually maybe encouraging these studies, but they have to be conducted first. So the researchers need to do these studies and need to submit these papers to us to give us an opportunity to highlight them. And there also needs to be a consensus within the scientific community that these studies are worth doing, and they're complicated, they're time-consuming, they're expensive.
Right.
Could I intervene for a moment, Christoph? I recall a conversation in one of the prep meetings we had here that it was noted that when you go to your peer reviewers, they tend to look at the science and go, yes, but what does it mean? What are the implications for therapeutics? They're not really focused on what does it mean for prevention, or what can we do to get to the point when people can stay well instead of having to fix people who have disease. That seems to be a bit of a cultural bar at the level of inside a journal and its process, I guess, with its reviewers. I don't know if there's any fix for that, but I found that an interesting observation when folks were preparing for this session.
Yeah, I agree. And I don't think there is an easy fix. I think, in many ways, we listen to the community and, we sometimes encourage and, promote these type of conversations, but there needs to be a bit of a consensus within the field that this is what we should be doing. So if these studies get funded and, we get conducted, then there is a chance, I think, for us maybe to put a spotlight on them and to elevate them and show that this is actually really important and can be quite meaningful.
Is there utility to special issues devoted to such topics, to prevention, to preventive metabolism, if you will?
I think as editors, that's one really good approach for us, just call attention to the issue. So we're listening to this meeting, so I could go back to my team and propose a forum piece on the topic. And I think, we aren't the main driver of the research, but we can boost it and direct attention to it. I think we all play a part in the system. But another thing I wanted to comment on with one of the hurdles that we see is sort of a need for more biostatistics training. So these big omics studies, these longitudinal studies, they're more difficult to analyze statistically, and we see that play out in the reviews. So if universities
just to make sure I
beef up these
understand,
programs.
You're making that call for action for whom? The authors, for the reviewers, for the journal?
I think that the funding institutes and universities just making a greater push for better biostats training, because I think it's it's a missing and very important piece of the puzzle.
So, I'm sorry, Georgina, go ahead.
It's okay, go ahead. I'm going to go.
Okay. So a comment was made a few minutes ago, and I hope I don't misattribute because I'm not writing anything down. I think it was made by George about, somebody dropping something on a dish of cells and ultimately making a therapeutic inference, which may or may not be cost effective, may or may not be practical, may or may not be mass producible, et cetera. And of course, the chain of events from a dish of cells to us is even longer than from black 6 mouse to us, of course. And so it brings to mind, I will go off on a tangent. There is a weekly email that is sent out by Dave Allison at the University of Indiana called Obesity and Energetic Offerings. And in that email, there's always a section called Headline versus Study. And it highlights 3, 4, or 5 headlines that have grabbed people's attention. And then underneath it says what the study actually said in brief. And of course, the study never says what the headline says. It said, is there a role for the journal, any of your journals or others, in getting that right? Or is that responsibility all on the journalists who get it wrong?
Well, I think, I'll start off because, at Nature, we are very cognizant that the studies that we publish are high profile. They will be disseminated widely and they will be scrutinized highly. And so that is something that is really built into kind of the publishing process at Nature. When a paper is accepted, we do an assessment as to whether this would be covered widely, and we have a PR team. And this person will write up a press release for journalists written in kind of a way that is accessible to someone who does not have specifically a biology background. And it really summarizes the, oh no. Can you still hear me? Yeah. It summarizes the findings and really states as plainly as possible their limitations. For bigger studies, we actually have press conferences. And so we invite journalists from around the world to call in, and we have the authors answer questions so they can really get an idea as to what the limitations is. I've sat in on a couple of these. They're quite interesting because you can see the journalists trying to see how far, most of the times, you know, they're corrected or their questions are answered. I think that this is, in this day and age, very important for a journal. Not all journals have the same resources. You know, this is a question of scientific communication and scientific responsibility, which I think is something that we don't really talk about a lot. I think living through COVID and seeing that discussing scientific findings in a responsible manner is can have huge implications for global health and for the trajectory of a historical pandemic. You know, this is why I think that bioarchive is something that's like very interesting, very helpful, but also needs to be taken with a grain of salt because those things are not peer reviewed. But in terms of, I would say, the media, getting back to kind of like your question, you know, there really is no group, there's no mechanism, there's nothing really to correct when a like Yahoo News article gets the study you just published wrong. You know, you can't like go through and say like Medline Plus like really got that study incorrect in their interpretation. You know, I don't know what to do about something like that. That is a big question.
Somewhat related. Sorry, go ahead.
Oh, I would reiterate what George said. It's a critical time. There's a lot of mistrust in science right now. When we meet with our PR team, the head of that, he's very conservative about like how we write things up. Unfortunately, even if we're very careful in composing the press release, what the journalists do with it, we don't have control. So I think there's a whole nother discussion on that and what happens down the line.
Sort of a related point in yesterday's session with Bill Hazeltine, lessons from the pandemic. One of the comments was the rise in preprints and how that was the speed of communication is directly affecting the speed of innovation. So they saw that as a good thing. Obviously, there's a balance there.
Yeah, but I mean, they call it breakneck speed for a reason, right? Like, you know, there is the speed, you can move things as fast as possible, and there will be absolutely communication of data. But the reason that the system is in a lot of ways built the way it is because there has to be a quality control process, you know, and that did not happen in those early days of the pandemic. And it is completely understandable about like the emotional state that everyone was in. Everyone was grasping for any information whatsoever. I think we all will never wipe down any groceries with like bleach or anything ever again. Hopefully we'll never, you know, but like it's one of these things where, you know, in retrospect, if we look then and now, you know, the scientific record has corrected itself the way it should. And now I think hopefully people are a little bit more skeptical when they're looking at preliminary data for clinical trials on medRxiv and bioarchive.
Yeah.
I'd just like to add here that I think that, the primary role of a journal editor here is to do their very best to ensure a robust peer review process and to ensure that the caveats and limitations that might have become apparent during peer review, that they are mentioned and discussed in the paper, right? Apart from that, the only thing standing between us and the publication or the communication with the wider public is maybe the press office, which we've talked about. And, we can do our best there. We're not press releasing every single paper. We typically pick those that we think would resonate with the wider public. But we sometimes deliberately also press release those where we suspect that the authors might oversell their data. Maybe we actually had to kind of nudge them several times to tone down their conclusions over the course of the peer review process. And, we feel maybe they'll just revert that when they press release. So we sometimes issue a very cautious kind of dull press release, which journalists would then have as a counterpoint to maybe a press release from the host institute that might sound a little bit overhyped. So that's one more thing we can do. But apart from that, I feel that the responsibility of how this is communicated to the public lies with the authors.
Very good. Thank you. I'm wondering if you have any further comment, Christoph, on the preprint servers. They have struck me as a double-edged sword. Obviously, speed of communication is wonderful, but the ability not only for the public, but even for the scientific community to sort through the best science versus the rest is, I think, a challenge.
Yeah, I agree. I think the preprint servers, they are here to stay, right? So preprints will remain a part and maybe an increasingly important part of how science is communicated in the future. I think the pandemic really pushed it to its limits. And, by disseminating the results early, this has really helped in some cases, and it was important to get them out as soon as possible. But I also feel that, the community was laser focused on making sure that the papers that are, wrong and might have been contextualized wrongly or overhyped by the press, that they are actually corrected, right? That's been pointed out. But that was because of the laser focus of the entire scientific community on a single issue that was of global, paramount global importance. That model is not scalable. So you can do it with a pandemic and maybe another health emergency that might come up, but it's not going to be, the papers will not receive the same level of scrutiny in all areas of science if people now start preprinting absolutely everything. So I think we'll have to live with the fact that they're not peer reviewed, and that scientists or the press might jump on them and interpret them in ways that were not intended to be interpreted, or they might simply be wrong. That's something that's going to be here to stay.
Yeah, I agree. They're here to stay. They're part of the ecosystem. A lot of people really like them. I think as long as we keep reiterating the caveats around them, that they're not peer reviewed. But it's interesting. There's still caveats around published literature until you've demonstrated that it's reproducible.
Yeah.
But coming back to the initial question that we all had, I know it's going to be very difficult to do preventive studies for chronic diseases, and I understand that. And I don't think there's going to be an easy way to do that. I mean, as we discussed in the prep meeting, it's very hard to actually target something and then see what's going to happen in 20 years in a person, or in a mouse. So how would you think we could tackle that? Maybe we can focus on some early metabolic changes, and then try to focus on that, so we can actually have an outcome that could be helpful. I don't know if I'm making myself clear, but I'm just trying to find something that we can focus on. And you three guys coming from excellent journals and also in the field of metabolism, I think we need to focus more on that.
I think they mentioned that the people have to do the research, and they're going to have to be funded to do the research, and then they can pass it through. Having said that, I'm hoping that conversations like this will help sort of sensitize what's the end here for society, and is there something, a role that the journals can play?
Yeah.
But I just want to ask the question, did any of you hear Luigi Ferrucci, the scientific director of the National Institute on Aging, yes, was it yesterday or day one, where he presented, and this is a very long period of study, so longitudinal phenotypic markers of aging. Those studies don't come along, once a week, but once you have results like that, I think they are important to disseminate. Do you see that kind of journal submission, or is that just a rare unicorn? And would you even publish it, actually?
Well, I can start. We are seeing more and more longitudinal omics studies, but they are rare. And it's really a matter of the field defining, what's meaningful across the span of that study. So you may do like lipidomics research and find lipid markers that indicate better long-term health. But what's the cutoff for that? What's a meaningful AUC? And then back to the mouse work, we don't even see that many mouse lifespan story papers, right? Because it kind of goes beyond the lifespan of a postdoc or a student to get that work done. So I think what's been really great in the field are these innovative, comprehensive markers of long-term health, like epigenetic clocks and frailty and disease. So I think at this point, it would be great to have, chair of science researchers come together and reach a consensus on like what are the most important markers and indices to look at, so that we can predict the future and we don't have to have 20 years of work.
Mm-hmm.
Correct.
Yeah, I mean, this is kind of the point that I wanted to make as well. Like, I think these studies suffer from the same problems that the field is suffering, is that we don't have good biomarkers of aging. So, it's probably not going to be a single biomarker, and we probably need a panel, but that needs to be agreed upon. It's not something that the journal decides, right? It's the scientific community who decides, like, these are meaningful markers that we should be looking at and following. So if anybody wants to get together and propose such a panel, that would of course be great. I mean, there have been, I think, really interesting publications, defining those sort of community standards and say, like, this is what we should be looking at going forward. With that type of dissemination, we can of course help as journals, but the scientific community needs to do, the groundwork.
Indeed.
Yeah, I mean, and if you take what I consider to kind of be the gold standard for long-term study, the Framingham study, that would probably be the ideal, where you not only have a population of people that are followed throughout their life, and that are phenotyped kind of, there's probably more blood from that city in freezers around the world than in the city right now. But it's one of these things where that's an incredible amount of work goes into maintaining and administering and registering and all of the things, and that's not necessarily feasible across the world for maybe offhand kind of like scientific questions. Yeah, what Rosalind and Christoph both said is incredibly important in terms of defining standards and seeing what the field thinks. I would say that in terms of these preventative studies, something that I feel like we run into with a lot of like the omics and big biomarker studies is just number, because I think we're starting to realize like the actual number of individuals that you need to include in your study in order to have something that is as robust as would needed to be able to be applied to the broader human population in terms of sex, age, ethnicity, all of these different things. And that, like I said, a large number of participants in any study is, anyone who's tried to register people for a study. That's a huge amount of work, money, time, and effort that not every institute really has. And so I don't know. I don't know if people should be working together more closely. If institutes that do have those resources should be sharing them more. That's something really that the field needs to figure out, because we receive at our journals, I'm sure between the three of us, so many papers that make claims about human health and use omic studies and biomarkers. But the thing is, how powerful are these studies to actually say something definitive that's broadly applicable? And that's why I think we reject so many of these papers, just because they are interesting questions. And you can find anything if you have a group of people and do lipidomics of their serum, but the question is it significant, and is it broadly applicable?
Can I ask a question from right field? I'm sorry, Michael. Just thinking, again, in an earlier session, there was a sense that a desire that regulatory science consider AI-generated discoveries, I don't know what noun you'd apply to them, insights, hypotheses, whatever, as a new form of doing regulatory science. What is your view about AI-based articles that are submitted? You're seeing patterns, that's nice, but in the regulatory field, you need to have them validated. And do you see those kinds of stories being submitted? And what's your view about when they constitute a sufficient standard, if you will, of doing science that you would publish them?
I can start. We've seen a few. I imagine George has seen more at Nature. I think they're hard to review because it's a black box algorithm. So it's really about transparency in the algorithm, how it was performed, letting the reviewers have access to the data. But it's fairly new for us, so I'd be interested in hearing what others have to say.
Yeah. So at Nature, I handle clinical cardiology AI. And so we do see quite a few of these studies, mostly in the scope of diagnostics, integrating typically for cardiac AI, integrating imaging data, echo data, these types of things, and now more and more electrocardiography data to make inferences about long-term human health and maybe incidence of disease in the future. But in terms of getting them reviewed, I mean, Rosalind is absolutely correct in that they're very difficult to get reviewed. A lot of these studies in terms of AI kind of fall into two categories, and it turns on really where the advance is the advance in a new advance in AI, or is it the application of AI that already exists to a biological question? And sometimes those two, a paper will have both of those aspects, sometimes it will just have one. And so really it's about, like Rosalind said, data accessibility. A lot of times these data sets are not accessible, and therefore reviewers can't even really probe the data themselves. There is a huge reproducibility problem in the AI field. And this, I think, goes more towards the fact that, individuals are taking these programs and using new novel data sets that they weren't kind of generated with, and seeing that they don't perform the same. I do think that in the next couple of years, I see the AI kind of reviewing AI papers is going to get more and more rigorous as the field realizes that they need to be more and more stringent, because it is a budding field. It didn't really exist, 20 years ago in the state, at least it does now. And so with more and more groups that also don't have a focus in AI using AI, that becomes a problem too, because then you don't really have someone who knows this black box and is just kind of like throwing it onto a problem. I find with AI studies, it's the intentionality is the most important thing. AI can answer a very, very specific question and not a broad question. And so when you try to start doing broad strokes with AI data, I think that's when you start to see the failure of the data to really hold up. Yeah, I don't know, Christoph, I don't know how many AI papers you've been seeing.
Yeah, I must say that the majority of the AI papers or papers involving machine learning that I see is in that those papers, they are more used as a discovery tool. So they might identify some patterns or, that are then subsequently validated. So they are just one part of the paper. There's usually interesting biology that's coming after it. So if the AI itself is the main advance, maybe because it's used in a diagnostic context, then that I think is much more difficult to review. And I imagine that it's also tricky because there might often be intellectual property involved. Maybe a company who, which developed such a clever algorithm would not necessarily want to disclose it in full. So yeah, maybe these papers never get written for that reason. So, and that could also be a reason why I have not been exposed to too many of those yet.
So would you like to discuss, all of you, because I remember we discussed this at the prep meeting with Rosalind, the current trends that you see in the literature in your fields? I mean, anybody can start.
Do you see any changes, any trends from the past 3-5 years to
what was like 10 years ago while you started your career? Well, 20, I don't know how many years ago.
Yeah.
Rosalind, go ahead.
Oh, sorry. I started in early 2016, so it hasn't been that long, but things, science is growing rapidly. I think I mentioned these before, really just taking the mouse work and putting it in perspective of human genetics. We're seeing so much more work in omics, lipidomics, metabolomics, proteomics, metagenomics. It's just sort of the data explosion, and that's the big challenge in how to analyze it, how to handle it, how to review it. So I think that's the biggest change over time. And the other one I've noted is, we talked about this a bit, like people are more proactively scrutinizing the mouse models and how they translate to humans. Do mice age the same way as humans? So we had a paper last year, I think, by Rafa de Cabo on just something very simple, fasting blood glucose. The trajectory in aging is very different between mice and humans. So we had to carefully think about how we assess that marker. And yeah, so sort of scrutinizing the mouse models, putting them in comparison to humans. And since I've been at Cell Metabolism, we've broadened our portfolio to include more human studies. Of course, we want to represent all branches of science. So, we'll still publish Drosophila, C. elegans, mouse work, but we've had a push to include more human work, and human diet and exercise studies have become more sophisticated. We're seeing more high quality RCTs, and that's probably out of the funding for them.
Yeah. What is it you wish you would see that you're really not seeing submitted?
I think, that is, I always say that some of the best studies that pass through my desk are the ones that I could have never anticipated. And that's just really the honest truth, the best science is the one that when you read it, you just absolutely know that this is something different and important and insightful. And it's something that, you know, people, it brings a perspective, it moves a field in a direction that was completely unexpected. And so that's why I actually think it's really not fair sometimes for editors to make wish lists, because my wish list is incredibly abstract.
Or surprise me.
Yeah, I mean, it's just, you know, between the three of us, we probably see most of the metabolism research that's done in the world, which is an incredibly privileged position to be in. And so we know what everyone's doing, and we know what's standard. So when we say something isn't standard, and then we get kind of like, there's a back and forth with the author, he's like, no, we know what's standard because we've seen 12 of these papers, you know, maybe in the past couple months. So we kind of know what people are doing, and we know what's standard. So when something new comes around as an editor, it really does, you know, it shakes you. And so that's kind of what we want to see. You know, a conference like this has set so many goals for where the metabolism and aging field should go. And as an editor, you want to see all of them achieved. You want to see all of these studies realized and published eventually. So yeah.
I couldn't agree more with what George had said, but there are maybe one or two things that I want to point out that I think I'd like to see a little bit more of. And one is actually, you know, because we do see a lot of mouse studies, right? And more studies involving female mice. I think sex as a variable, right? It's really important, and it's something that has become a bit more important now, and people paying more attention to. At the journal, we've sort of started a trial where we're actually enforcing a little bit more the reporting related to sex and gender in our papers. I must say that the aging field is actually pretty good at that, at least when it comes to survival curves. You typically see male and female mice, but a lot of the metabolism research is still just being done in male mice, purely because the female mice, they don't get quite as obese on a high-fat diet, and therefore they're excluded, or, you know, there are no really good models for fatty liver disease or NASH in that work in female mice, and that's I mean, that cannot be right. The science needs to do better than this. So that's one thing. And then personally, I feel when it comes to aging research, I'd like to see, I wonder whether we, you know, shouldn't be doing more experiments in old mice. Because the tissue microenvironment is very different in old animals. The nutritional requirements of an organism change with age. You know, how disease processes unfold in old animals and old people is likely very different than in the accelerated disease models that we typically use as a shorthand. Of course, they have their place, right? But you know, we're coming back to this other problem that the time scale to do these experiments is very long. And maybe this is actually another point for the discussion where we have to think about that the timescales for publication and, you know, conducting research and publications are now sort of increasingly uncoupled from the time span it takes to do a PhD or for funding cycles, which is a problem.
So, you know, this has been a bit of an experiment to see what would happen if we got editors together. And I've actually learned a lot. And it's interesting to hear this discussion. We've reached the end of our time. So,
but I have several more questions that I wanted to ask.
So let's keep the conversation going offline. So thank you very much. And I think I'm going to ask Zan, our co-chair,
We're
to come
looking
back.
forward to your comments about the conference and in our survey.
Please.
Sure. Thank you very much for having us.
Thank you for
so much.
participating.
Thank you so much.
And we are going into a break, so Zan's going to talk a little bit about that. Please come back at 10:45 for the commercialization.
I did want to say to our panelists and to Michael, bravo. I just, you know, I was really losing some sleep about how this panel might go. It's a high wire act to bring people together in a kind of first setting. And I could not have dreamed that it would have gone so well. So congratulations.
We embrace experiments.
Yeah, and we sometimes fail, but this is a labor of love, and we do this on a unbudgeted basis. We're not professional conference producers. We have some professionals who try to help us do that.
Not let us fall down.
But yeah, we don't have a net, and so we beg your indulgence from time to time. But
It's exciting.
gosh, that was a world-class discussion. We can't thank the panelists enough for that. As you say, Thomas, great insights.